Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last few classes of this week, we have talked about principal component analysis, which is a linear dimensionality reduction technique. We have talked about very briefly about the theory of this PC analysis. We have then done hands on, and we now have got a fair idea of what principal component analysis does and how we can interpret the results of that analysis, how we can extract meaningful information for further analysis. In this class, we will be talking about a nonlinear dimensionality reduction technique, which is called the UMAP. And very briefly, I have mentioned UMAP, but I will go into a very brief discussion of what is UMAP analysis, and then we will do the hands on with the R package UMAP. Okay, so this is something that is the agenda for this class. Very briefly, I will first remind you about UMAP analysis. As I told you, this is a nonlinear dimensional reduction technique. So, what it means is that very similar to principal component analysis, we will also have this UMAP 1 and UMAP 2, right. So, when you do the plot the data, we will see this that reduced dimensions, we have UMAP 1, UMAP 2, but unlike principal component analysis, we will not see any loadings of UMAP 1 or UMAP 2. Okay. So, this is a very important difference. What it means, this method will not be able to tell us which features are the most important ones. It will be able to show us the structure of the data, right, from a very high dimensional data. It will generate a low dimensional data or project the data in low dimension, which will give us a idea about the structure of the data, but it will not be able to tell us the most important features which PCA did. But this is also important because not all features that we use are will have linear combination, right. So, the final principal components, they are not always final principal component will be a linear combination of the features, whereas UMAP is not, and this is something that is true for biological data, right. Not all features are linearly correlated or have linear association. So, this is a nonlinear dimensionality reduction technique, and it uses something called graph layout algorithm for data representation, and the goal is to represent the data, high dimensional data, in a low dimensional space, so that the data closely resembles, right, the data in the high dimensional space, but and also retains the most important information, right. Most of the structure should be retained. Okay. So, that is the goal, and we have also talked about very briefly that the two important parameters that allows us to kind of change, right, whether we want to retain the local structure or the global structure, that kind of information, right. This is something we have control over. So, we will move on to the hands on, and we will use the cancer gene expression data set that we utilized for the first principal component analysis, and we will use that data set for UMAP analysis, and again we will do some visualization, and you will see the difference now, right. When you do the visualization here and what kind of output we are getting from UMAP, right. Then so you can go back to the principal component analysis on the same data set, and you can compare, right, how UMAP differs from principal component analysis. So, let us now jump into the hands on part, and we will first load the data set, and then we will also look at the package. Okay. So, the data set we are familiar with, and here is the 100 genes PCA2 new. This is the data set that we use for principal component analysis as well, right. So, we will load that data. We will also have the labels data set, labels.csv, and then we will do some specific pre-processing that we did before, and then we will continue with the UMAP analysis. For the UMAP analysis, we will use this package UMAP, okay, in R. This is something you can download. This is in R package. So, you can simply say install.packages, and this package will be installed. Here is the reference manual. You can check, right. Here we will have the functions that we can use, right. So, the functions that we want to use is this UMAP, right. So, this is simply we will calculate this UMAP for on the data. The parameters that we have to provide, right. We have this D matrix input data. We can say configuration, right, UMAP defaults usually, right. The method, right, again this is a different method that is in R, right. So, you can use naive method or UMAP learned. So, this is something, again, this can be specified, but we can just go with the default which is written for R, because if you want to use this UMAP learn package, you should have that installed in the system. Okay, that is a package that is actually a Python package that we have to independently install in the system. Okay, so this is how it's a very simple and a small package that we can use, and here are some probably examples, right, that is kind of given here, and this is something what we are going to use, right, the UMAP package, and then we'll visualize the results. Okay, the data set first, so starting with that, we first load these packages, right. We need the UMAP package, and we also use this tidyverse and dplyr for data organization, data transformation. As we have seen, these are very, very useful. The data set that we are going to use is this original data read.csv, right, because this is we are reading a CSV file, so we need to use this read.csv command here, and we have seen this data set before. So first we can look into the dimension. So again, 10,000 samples, we have 104 columns, so 103 genes and 1 label, right. So we can simply look into the names, right. Okay, so as you can see, these are the gene names, right, 103 gene names, and the last column is the label. And if you remember, this label is for the cancer type, right, that we are working with. The row names we can see, of course, so we do not have row names here, because the label is given. So we can simply look at this, right. We have genes and these are the samples. There is no sample name, but the labels are given. This is kind of describing the cancer type. Okay, so we have the data label file as well, and which we can load here, data labels read.csv, so labels.csv here, and as before we can simply look into this unique original data. So we have labels 0 to 30, right. This is something we are familiar with. The other thing that we did also last time is we can do this original data group by label and count label, right, how many samples we have for each cancer type. Okay, so here for label 0 for this cancer type we have 173 samples, for the next one we have 79, and so on for 31 cancer types in our data set. As before, we will not be working with the full data, because that will take a lot of time to actually do the UMAP analysis. So we want to kind of demonstrate this idea how to do this with a small data set, but then of course you can learn this on the full data set and see what kind of results you get. So for doing that, we will choose this data new. Okay, and what we are doing here as before is we are taking this 1 to 50, right, first 50 level 0, 174 to 223, so next 50 we have from level 1, then 253 to 302 from level 2, and so on. So this will give us the only first 3 levels, right. As I said, this is a huge data set, we do not want to take the whole data set for our analysis. So we have taken only about 150 samples, right. So we have 150 from first 3 cancer types. Then we are taking these columns, right, where we have this first 50 columns, we are taking only 50 genes, right. We are not taking the full gene. Again, for we want to minimize the size of this data set, and we also are taking this label column, right, where we have the label information. Okay, because we can use this label information to actually do some plotting and visualization. What we can also do is we can replace these labels now, and we can actually put the cancer types, right. Here we know the cancer types now. So instead of 0, 1, 2, we can simply say LML, SSC, and DLC, right. These are the abbreviations for these cancer types. You are familiar with this command, we have used this before. This is replicate or repeat, this C, right. So these 3 each 50, right. So you will have 50 of these, then 50 of these, then 50 of these, right. This is how the sample is stored. So we get this label now. Okay, so we can look at this data new, right, and you can see this, right. These 50 genes are here now, and the last label column is retained, and the label is also now replaced from 0, 1, 2 to actual abbreviations for the cancer types. Okay, so now we are ready with the data. We can also generate this data new, right. From data new we regenerate this. We do not want to include the label for UMAP analysis, right, because this is something that is not a numerical variable, right, that is not kind of continuous variable, this is something just a label or class. So, we do not want to include that in our UMAP analysis. The other thing we also would define here is these data labels, right. This is the data new label as factors. Okay. So, what happens when you say define as factors is like this is the list that we get. Okay. So, and it will tell what are the labels that we have. This is something we have probably discussed when we are talking about different data types in R, right, in the first few classes, and this is we can define a list of values as factors, right. So, this is what we have done, and this is something we can use later on. Now, how do you run the UMAP is very simple, right. We can say UMAP data. So, the function is UMAP as we have seen in the package manual, right. So, we can simply run this UMAP data equals to UMAP data, okay, because it is a small data. So, this is done now, right, and let us see what we have. Okay. So, it is kind of like just saying, okay, we have 150 items in 2 dimensions and we have different components, layout, data, gain and config. Okay. We will see what we have here. What we can then next look at is summary UMAP data. Okay. So, this is what this is showing, right, summary UMAP data. Okay. And we can also read this a bit, right. So, layout length is 300, data points is 7500. Okay. Why? Because we have 50 genes and 150 samples. So, you can see the dimensions of that matrix, right, and UMAP embedding of 150 items, these are actually the sample types, right. So, we have 150 cancer samples. So, 3 cancer types, and this is UMAP embedding in 2 dimensions. So, we will see this plot. When you plot this, it will be UMAP 1 versus UMAP 2, and we will see the data points. Okay. And then we also have UMAP KNN, UMAP config, etc. So, this is something we will not go into too much right now. So, the other thing we can look into is the UMAP layout, right. This is how the layout looks like, right. These are the values of these samples. For each sample, these are the values, right. When you plot these xy coordinates, right, so UMAP 1, UMAP 2, this will be the xy coordinates, right. So, what you see, like from 50 genes, this is now have been reduced to 2 dimensions only. Okay. And we can simply call this plot UMAP data layout, and this will generate this kind of plot. Okay. And you can probably realize that these are actually the 3 cancer types, right. We can guess, okay, from this visualization that these 3 clusters, they represent 3 cancer types, but can we now color them and kind of see that clearly, right, because we will not always have 3 cancer types, we might have a more, and they might not be so well separated, and this is something we will do in the next part, right, plotting part. Okay. And this is how we will go about it. There are 2 ways we can do this, right. One is we can simply define this color set of colors. Okay. Okay. Yeah. All right. Let us see. Okay, so we are defining the colors here. Okay. So, what I am doing is very simply I am taking these 3 colors red, blue, green. So, that we will cluster this in red, blue and green colors, right, 3 different clusters, and each equals to 50, right. So, for 3 cancer types we have red, blue and green color, and then each will be 50 because we have 50 samples, right. So, this is how we are defining the color variable, okay. And then what we are doing is we are using the same function, right, but there is an important difference here, which is color equals to color, right. So, this is means we are going to use these colors for our data points, and PCH equals to 20 and the point size, right. So, what will be the point size? Okay, so here is the plot again, right, and we can make it even nicer by changing the labels, etc., and size of the labels and so on. But what is important is to look at the data points, right. So, what you see is for red we have a very nice cluster that is kind of well separated from the rest of the blue and green points. We have green cluster that is kind of pretty good, right. It is again a very nice cluster. In blue cluster, however, we see some green data points, right. So, green points are actually coming with blue cluster, right. So, which means some of these samples might have some similarity with this blue cluster samples, okay. So, this is something that we see, and what you also notice from our comparison with principal component analysis is that the separation is much larger here, right. So, this is because of the algorithm that is used. So, you can see these clusters are now very well separated and kind of they kind of far away from each other, but also we see some green points within blue points. If you remember in PCA, and if you go back to that class, you will see that they were actually much closer in principal component analysis. Now, this is something that is very important to remember, and we have discussed when we are talking about principal component analysis and UMAP, is that the distance in UMAP is not relevant at all, right. So, we cannot actually use that information for any purpose, whereas in PCA that information is important. The distance actually tells us, right, how similar or how dissimilar the samples are. Okay, so, we have now generated this plot, right. We can separate out this, and we can also make this a bit nicer, okay, with these labels, right. We have UMAP 1, UMAP 2, these are the axis labels, right. So, -4 to 4 range. We can also use legend function. This is something we have not used yet. In R we can use this legend. So, we will use a legend on the figure, and it is saying the location of that legend, right, whether we want here, bottom left, bottom right, top right, and so on. So, we can go for top right now, okay. All right. So, we can go for top right because this place is empty. So, we can put the legend here, and the legend is simply these cancer types, right. So, this is the LAML, SCC, BLCA inset, right, the location, color, what color they correspond to these labels. So, red corresponds to LAML, blue corresponds to SCC, and green corresponds to BLCA, right. And this is what we are going to put there, okay, and we do not need any border and so on. So, this is something that we can put here, right. So, this is something we generate the full plot, and we also add the labels, and then we kind of know, okay, these are the LAML samples, these are the SCC samples, but some BLCA samples are along with the SCC samples, right. So, they might have some similarity in terms of expression patterns, and here are the BLCA samples that are kind of separate, right. So, this is something we can do, but there is another option that you can use for this is using this library WGCNA. There is a function called labels to colors, okay, and this is something we have used before, and here we can also use. So, this will automatically convert labels to colors, right. So, this is what we are doing, right, and we have defined these colors brown, turquoise, blue. So, this is a default kind of automatic conversion. Then we have the plotting colors. We take these colors and replicate each of them, right, 50 times because we have 50 data points, and we then plot this UMAP data layout using these colors, right, exiting the same way, right, and again we have very similar results, and we can add this legend here and maybe at top right again, right. Okay, and we have the legend, and now we can read, right, which data points belong to which type of cancer. Okay, so this is something we can do. All right, and the next thing I will describe, right, we can actually have a you can also have a generate plotting function that is actually is also from UMAP, right. So, in this UMAP package there is this somewhere, okay. So, maybe I will just open that link and it is part of that package somewhere. Okay, so part of that package and here in implementation, so you can see again there is some examples of this package, etc. But what you want to look at is in this package below we will there is a custom plot function that is also given, right. So, this is a function that can be used to actually plot the results of UMAP, right. So, this is something we can use, right, and we can try here, right. So, what you need to do, so this is the function that has been defined in that link. So, as part of the UMAP package that is actually a custom function that has been written, we can take that function and we can do the visualization of UMAP analysis. Okay. So, as you can see, this is a function. So, this is in general form which will take this UMAP data, right. So, this is the UMAP data after running the UMAP function. Then we have the data labels here. So, these labels are the labels that we have generated, right. You can see this, for example, right, these are the data labels as factor that will be required. So, some of these things are important because you will see, like, if you do not store them as factor, you might not get, you will get error, right, in many of these plots. Okay. So, what we will do is, okay, so here is the labels that will be taken, then this is the main colors, red, blue, green, you can define any colors that you like, and then you have different parameters for aesthetics part. Okay. And then what we have is something that you probably want to look at very carefully, what is actually happening. It is kind of like automating the full analysis rather than so for any UMAP analysis that you do, we can call this function and we can generate a plot. Okay. So, here is a layout that is kind of being defined, right, is x is a UMAP, then there is this is the layout that will be used, then you have the xy limit, right, that is the range of the layout, right, what should be the limits, etc. Then you have the labels function, right, this is a labels function, that means again taking the labels that are there, and the legend position is default, by default is top left, and of course, this is something that we can change in the custom function. We can go and change this, we can say top right depending on the appearance of a plot, right. And then we have the labels, right. So, if you are doing this as legend also, right, you can see, so labels you have, then you have the legend position, right, and legend text, and then you have the legend given, right. This is something defining this legend position, legend text, etc. Here is the legend function, right, very similar to what we did, but this is more kind of custom function which you can call simply by saying the UMAP data that we have done and the labels of data labels. Okay. So, we can do this. Okay. First, we have to load this plot function in R, right. This is an R function, we have seen how to write these R functions, right, and then we can call this plot function UMAP data labels. Okay. And as you can see, here we have the legends, okay, and here are the LML, SEC, PLC. Of course, we do not see the axis, but in any case, as I said, the distance between the clusters do not matter in case of UMAP analysis, right. So, this is something that we kind of do, and we kind of see these kind of clusters. We can try this for a bit more data set in our case, right. So, we have done this for a small data set, maybe we can see for maybe a few more, right. So, what I will do is let us run this again and we will choose some of these data sets, maybe we can choose 50 of each type, but then also some of the other labels that we have. Okay. And we can also see the labels that we have. Yeah. So, using this data labels function. Okay. Okay, so, this data labels is actually coming from the labels.tsv. So, Okay. So, this what we have is 0, 1, 2, 3, 4 and so on. So, maybe we can take 3 more classes like LGG, breast cancer, CESC and all these 3 different cancer types and see how this data set looks like. So, that will be much more complex analysis, right. We can try that, and so let us go back here. So, what we have, let us try with level 3 LGG and then level 4 BRCA. So, level 3 we have about 530 samples and level 4 we have 1218 samples. Okay. So, let us choose some of these samples now. So, for doing that we will have to see where we are. So, where it will start. So, 173 plus 29 plus 6, so 678. So, we can choose 679 and then 50 samples, right. Okay. And then we have plus 530. So, we have 1208, maybe 1209 to 1259. Okay. So, let us see, and maybe we can choose a few more genes, like maybe 60 genes, so that we get bit more information, right. So, as you increase the number of genes in the data, number of features in the data, it becomes more and more informative, right. So, you can probably see better clustering in most cases. So, we have LGG and we have BRCA. So, we have to define this now. Okay, so maybe it is not matching to some extent. Let us see, I think we have to consider. Okay, so this will be 1258, right, 50 samples. Okay, so we have this now, head data, okay, and the data labels we have like this. Okay. So, this is the labels we have defined as factors, right. So, we now have 5 labels. So, 5 factors, right, 5 types of cancer data here. Similarly, we can run this UMAP data. Okay. So, this is all right, and then we can simply plot this. And here it is an interesting part, because now UMAP 1, 2. So, we had 5 cancer types, but we see kind of here probably 2 clusters are merged together. So, we can again now color them, and we can use this color. So, because we do not need to define these colors ourselves, we can simply say labels to colors using this function from WGCNA package. And okay, so we have to move the label maybe to the top left. Let us see. Okay. All right. So, we have moved the labels here. You can see. So, this yellow one here is the LML. Then we have ACC here. We have BLCA here, LGG here, and BRCA here. Right. So, you can see these 2 clusters. Okay, they are separated out very nicely, but they are close here. But some of these blue and brown samples, they are kind of mixed up with here. Okay. So, this is something that we can do, and you can see now clusters are also tighter because we have also introduced a few more genes. So, if we have more information from which UMAP can generate these clusters and do this dimensionality reduction. Okay. So, we have now done the hands on for UMAP. So, let us go back to the presentation. And so, here are the references for UMAP package. So, to summarize, what we have done is we have talked about UMAP analysis. This is a nonlinear dimensionality reduction technique, and this kind of projects the high dimensional data into low dimension and trying to keeping the structure of the data more or less intact, right, and trying to preserve information as much as possible. This is something that is very helpful for looking at the patterns in the data, the global structure as well as the local structure. And this is something which we have seen now with hands-on example, where we use the package UMAP and we have done the hands-on analysis on a cancer gene expression data set, and we have seen that based on these gene expression patterns, UMAP could cluster these different samples, right, and we got these different clusters for different cancer types. And then we of course did the visualization, which is a very informative and important part of this kind of dimensionality reduction techniques, because this kind of gives us an insight into how well these samples are separated, or whether there is some sort of similarity between samples or not. So, together we have now talked about the linear dimensionality reduction technique, which is the principal component analysis, and we have also talked about the UMAP analysis, which is a non-linear dimensionality reduction technique. We have talked about their advantages and drawbacks, and we have also done the hands-on analysis, so that now we have a fair idea of how these analyses work and what kind of results we would get and how to interpret those results. Thank you.